Now we are going to take the next application of biotechnology in medicine, in the field of medicine. That is the second one is recombinant human growth hormone. First we will take growth hormone, which is secreted in our body. If it is deficient, what will happen? Then how the hormone is synthesized with the help of biotechnology. We are going to study. Say all the human beings are having a gland below the human brain. Okay, below the human brain. Now this is the human brain, side view and taking. This is the front, this is the back. Below the human brain, there is a small oval-shaped gland. This oval-shaped gland is called a pituitary gland. This gland is called a pituitary gland. Now the pituitary gland is having two lobes. One lobe is very bigger. It is called the anterior lobe. It is called the anterior lobe. The next one is posterior lobe. Okay, the next one is posterior lobe. Okay. But I am going to take this anterior lobe. The anterior lobe is secreting 5 different types of hormones. Not 1 or 2, 5. In that the first hormone is called growth hormone. Okay, this hormone is called growth hormone. Abbreviation is GH. Growth hormone, GH. This growth hormone is also called somatotropic hormone. Okay, it is also called somatotropic hormone, STH, another name for it. Now, if this is a somatotropic hormone or growth hormone is secreted normal from the childhood, the child is growing in normal height. For example, we are all normal in height because the growth hormone is secreted in normal quantity. Rarely for some individuals, this growth hormone will be secreted less quantity, insufficient quantity from the childhood. So the child cannot grow normally. The child is showing dwarfism. What is that term is? Dwarfism. The dwarfism means the individual is abnormally shorter. Age is increasing for the child, but the height is not increasing. Now for the childhood, they are coming to the teenage to adult. But even if they are reaching the age of adult, 20, 21, 22, 25, 30, their height will be 1.5 feet to 2 feet. They are not normal in height. That condition, abnormally shorter condition, is called dwarfism. In the society, human society, rarely some people are present who are very, very short. They are called dwarfs. They are called dwarfs. Very short people. That condition is called the dwarfism. Now, if it is early detected in the childhood itself, if we inject the growth hormone to the child who is having less quantity of growth hormone, the child will grow normal height like this. So, to avoid this dwarfism from childhood, we have to inject growth hormone. Even here also the growth hormone, the old method, it is very, very difficult to extract the growth hormone either from the anterior lobe of pituitary gland or from the blood. It is very difficult. So now we are applying this biotechnology, and by using this recombinant technology, we are synthesizing. Synthesizing means manufacturing human growth hormone artificially. Whenever it is coming biotechnology, it is artificially we are doing this. Once again, I am going to take the same way. Now, for the growth hormone from the anterior lobe of a pituitary gland, we have to take one cell. Now, that is nucleus. In the nucleus, chromosome I am taking. In the chromosome, there is a gene. This gene is meant for growth hormone. As in that, it is only responsible for the production of growth hormone. But the gene is defective. Now this is the gene, but the gene is defective. It is not activated properly. So the gene is not preparing or not producing sufficient quantity of growth hormone, as a result of which dwarfism is caused. Now we have to isolate this gene for growth hormone. Same way, we have to take one E. coli bacterial cell. Now it is having its own circular chromosomal DNA. Then apart from that, it is having a plasmid. Now already I told you the plasmid is a special DNA which is present in Escherichia coli, E. coli bacteria. Okay, this is a plasmid. Now this is the gene which is taken from the human body cell for growth hormone. After that, this is the gene. Okay, this is the gene which is taken from the pituitary gland cell. Okay, this is the gene for growth hormone. Just we have to incorporate this growth gene, hormone gene along with plasmid. Now once it is incorporated, it is called recombinant DNA plasmid or recombinant plasmid DNA. Now once the human gene enters into the bacterial cell, we have to make the cell to multiply in thousands and millions. Then once the gene is attached here, in the cytoplasm, another one hormone is produced. This hormone is human growth hormone. It is secreted. How much it is possible? It is secreted. Now what we have to do is, we have to extract the human growth hormone secreted by the Escherichia coli, and you have to purify it, and that could be given as injection. Human growth hormone injection. It is artificially prepared. It is artificially prepared. So this can be given to human being, particularly the child which is showing this problem of deficiency of growth hormone. If it is substituted, the child is growing normally, the dwarfism will be avoided. So the person will become normal. This is the second technique in the field of medicine. Now we are going to have the third technique. We are going to have the third technique. Now we take what is actually this third application. We are going to see in the field of medicine. In the field of medicine. So we are going to study about human blood clotting factor. Okay, this is called human blood clotting factor. Human blood clotting factor. So first we have to know what is this human blood clotting factor. See, in our body, blood is there. It is red in color. You know the combination of the blood. It is having a fluid chemical called the plasma. Then it is having three types of blood cells: RBC, WBC, platelet, everything. Now our body is having a peculiar property. What is the peculiar property means? For example, we have a small cut on the finger. A small cut, not very deep. Blood is coming out. From the injured part, cut part, the blood is coming out. Now if you leave it as such, after some time, the fluid blood on the cut region is slowly changing into solid factor, which is covering the cut portion. After that, blood is not coming. It is called blood clot. So blood clot is an important property of human blood. Otherwise, wastage of blood will be present. Say for example, whether it is a small injury or a deep injury, when the blood is oozing out, the clot, that is the fluid blood, is changing into solid blood. For example, I will take it here. This is the skin surface. Okay, here there is a small cut. Okay, this is a small injury. Now here the blood is coming out. Okay, the fluid blood is coming out, is flowing out. Blood is very valuable substance. It is coming out, but within half a minute to 1 minute, the liquid blood is converted into solid substance which is covering this injured part. Now this is a solid substance. This is called clot, blood clot. Say for changing the liquid blood into solid substance, that is for blood clot, one chemical is important. It is called blood clotting factor. The name of the chemical is called blood clotting factor. If the blood clotting factor is there, blood clot will take place. Imagine that in one person, the blood clotting factor is not present. For such an individual, this is the skin, damage is there or injury is there, the blood is oozing out, the blood is coming out, but the clot is not taking place. So the blood is continuously coming out. This defect is called hemophilia. Now hemophilia is nothing but continuous oozing out of blood, continuous flowing out of blood, no clot. So what will happen? Within 5 minutes to 10 minutes, heavy amount of blood will come out of the blood vessel, come out of the body, so the person will be fainted. Further, if it is not clotted, what will happen? lot of blood will shed off, then the person will die. So this lack of blood clot and continuous oozing out of blood is called hemophilia. It is a disease. It is a disease. Okay, how to cure this disease, hemophilia? We have to give the blood clotting factor injection. We have to give. Then only the blood will be clotted. Now, for taking the blood clotting factor, the chemical from the normal human being, we have to take lot of blood from the body. Maybe about 2 liters of blood for taking 1 ml of the blood clotting factor. 2 liters. Nobody can donate this 2 liters of blood. So, from that. So, it is a wastage and also practically it is not possible. So, we have to call 4-5 people for giving this 1 ml. But by giving 1 ml it is not enough. Periodically we have to give 3 times, 4 times, 5 times. Then you can imagine how much amount of blood will be needed. Even for this, what they are doing is the human blood clotting factor, they are producing artificially by using this biotechnology. So what they are doing is, they are taking this cell, human body cell. In the human body cell, the same way, in the nucleus, in the chromosome, there is a gene which is responsible, this is a gene. This gene is responsible for the production of blood clotting factor. Okay, blood clotting factor. Now this gene, we have to isolate it and we have to introduce this gene into the ovary cells of ovary. We'll take it like this. Cells of ovary of, what is that? I told you that animal, hamster. The name of the animal is a hamster. Just before I told, hamster is not rat. It is like rat, giving appearance like rat, but it is a slightly bigger. It is a hamster. In the female animal, in the cells of ovary, we have to introduce this one. So the cells of ovary of a hamster animal will secrete more amount of blood clotting factor. Okay, blood clotting factor will be secreted. So we can extract and we can purify them. Suppose the female animal is not available, then what we can do is, we can introduce the gene into the cells of kidney of young hamster. Okay, young hamster. That is, all animals are having kidneys, whether it is a male or female. So in the kidney cells, we have to introduce the gene. So the kidney cells will produce the blood clotting factor. So we can extract it. So once we extract it, we can purify it and we can preserve it, and the injection can be given to any hemophilia patient. If they come to doctors, come to know that the person is having continuous bleeding, immediately they will give injection. So immediately the blood will clot, blood wastage will not be there. There is no risk for the life of the person. Now this is the technology what we are using. So that is the third application, that is human blood clotting factor. Next we are going to study about another important application of biotechnology. It is for interferon, one chemical substance called interferon. Okay, now we are going to take about interferon. The interferon is commonly called antiviral protein. It is protein, like our nucleus. It is a protein, but this protein is acting against the virus, so that it is called antiviral protein. Normally, we are having immunity, resistance. Now, the resistance is due to the presence of antibody. Our blood is producing antibody. But how the antibody is produced? That is with the help of interferon. That is with the help of interferon to some extent. I will tell you some importance of this interferon. How it is produced, how it is acting against virus. It is acting only against virus, not against bacteria or any other microorganism. Okay, now I will take our body cells. Normal body cells. Okay, everywhere body cells are there in the whole body. Body cells are there. These body cells are getting nutrients because we are eating food. Food is digested, absorbed, and it is going to each and every cell. So our body cells are receiving lot of nutrients. Okay, carbohydrate, proteins, fat, vitamins, minerals, everything it is getting. The body cells are growing. Then in the body cells, one special chemical is synthesized. The special chemical substance is interferon. Interferon. So we are not consuming this interferon along with our food. We are taking normal food, but it is digested and absorbed by the cells. After getting the food material, the cells are preparing a new chemical. That new chemical is called interferon. All the body cells are producing interferon. Now what the interferon is doing, once it is present sufficiently, it will go to the blood. The interferon will be present in the blood. So in the blood, if any virus is entering inside, a virus is very poisonous. It is going to produce disease. 100% it is sure. But what this interferon will do, the interferon present in the blood will come and make the virus inactive. The virus cannot be active. It is inactive. Its activities are suppressed. So the virus cannot multiply. The virus cannot produce poison. The virus cannot produce any disease at all. For example, interferon is there in our blood sufficiently. Now one virus is entering, maybe group of viruses, maybe 10 viruses are entering, same type of viruses. They are ready to cause diseases. But once they enter, the 10 viruses are inactivated by the interferon. So the 10 viruses will be present as such, but they are keeping quiet, they are not active, so they cannot produce any disease, they cannot multiply, they cannot produce any poison. So they may be present for some time, after that they automatically die. This is the importance of interferon for any normal individual. But for some individuals, this interferon, the body cells are not secreting interferon. Why? What is the reason? Say for the body cells, in the body cell, one cell I am taking, that is in the nucleus, in the chromosome, there is a gene for interferon production. There is a gene for the synthesis of interferon. But this gene is, what is that, we can call it as, it is very weak. The gene is very weak, or the gene is inactive. Okay, so as a result of this, the interferon is not at all secreted. So what they will do, they will take this, gene, okay, they will take the gene and they will introduce the gene like previous one into the microorganisms like bacteria. So the bacterial cells are synthesizing, manufacturing interferon. Okay, interferon. And this interferon, once it is synthesized, it will be extracted from bacterial cells, and the extracted interferon will be injected into the person who is having less immunity, into human body. So once it is entered the human body, then this interferon will mix with the blood, activating the, inactivating this virus. So the virus will be inactivated. So actually this is the basic concept. So about interferon, antiviral protein. But the interferon, if you take in the human body, the cells are producing interferon. There are three types of interferon. One is alpha. The second one is beta. The third one is gamma interferons. Okay, interferons. Three types of interferons will be there. One human individual may have all the three types, or any two type, or any one type. But that is enough to suppress the activity of viruses. So, any type of interferon can be artificially manufactured with the help of bacterial cells. This is actually interferon. Now we are coming to the next one is recombinant vaccine. We are going to study. It is the last technique. What we are going to study is the field of medicine, vaccine. Recombinant vaccine. So first we will take how we are manufacturing this vaccine and how these recombinant vaccines are prepared in the modern days. In modern science we have to study. Now first I will think about how vaccines are prepared long time before when this has started like this. Now I will take one example for this polio vaccine. How this polio vaccine is prepared. One example I will take. Say the polio is a disease. So normally the disease is caused by virus, polio virus. This polio virus is easily attacking the children. Less than the age of 3, easily they are affected. Once the polio viruses enter into the body, they are multiplying. After that, the polio viruses are damaging the nerves of the hands and legs. So the hands and legs are becoming paralytic. So the individual is having paralysis. That only we are calling it as polio disease. So for this they prepared a vaccine. Nowadays we can find in the media, social media, that is a polio vaccine, mass vaccination throughout India. One particular date they will fix up, on maybe Sunday, Saturday like that, and they are giving vaccine for all the individuals, either in the form of injection or through the mouth, the vaccine will be given. So how they prepare this polio vaccine? To eradicate this disease polio, now it is almost eradicated. How it is prepared? Now we have to collect the polio viruses, very dangerous viruses, very powerful viruses. And what we have to do is, the old method I am telling, these polio viruses should be inactivated. Okay, this inactivated means we have to make the powerful polio viruses into powerless. That is called inactivated. These inactivated viruses are also called attenuated viruses. We have to make it. Now, once the viruses are inactivated, even if the inactive viruses enter into our body is not going to be affected by the disease called polio. Then this inactivated virus will be preserved in the preservative. It will be preserved in the preservative. This preservative plus inactive vaccine together we can call as polio vaccine. Suppose a polio vaccine is a given to a child. The virus is entering. The actual virus is entering. But the virus cannot cause disease in the child's body because it is what? Inactive. They are unable to do, they are powerless. But once this vaccine is entering into the child's blood, the child's blood is producing antibody. Little by little the antibody will be produced, so that they are giving periodically the polio vaccine. Now once the antibodies are produced sufficiently, after that through the air or any other way, if the powerful polio viruses are entering, the antibody will destroy the powerful viruses, polio viruses. Powerful polio viruses will be affected, will be destroyed immediately. So the powerful polio viruses will not have the chance to damage the nerves. So the individual, the child, when it is growing, paralysis will not be caused. Now this is the old method what we are seeing so far. Now we are going to see how the recombinant vaccine is prepared. We have to see this. Now we have to take vaccine. That is, this is the virus. Imagine like this. This is the virus, and in the virus DNA is present. Now one particular gene is actually the gene that is polio virus. Now we have to isolate the gene. Now this is the gene of the polio virus, and this polio virus is producing polio. What is that? Poison. Okay, it is producing poison. That poison only is spoiling the child, nervous system of the child. So what we have to do is, we have to take this DNA, small segment, and the DNA should be suppressed. The DNA fragment. Okay, the DNA fragment. It is otherwise called the what? Polio gene. Now we have to suppress it. What we have to do? The fragment of DNA, by applying certain technique, the gene should be inactive. Okay, the gene should be inactive. Artificially we are making it to become inactive gene. Now this inactive gene, once it is introduced and the gene DNA, okay, that is an inactive gene, otherwise called inactive DNA. Inactive gene, I will write it, inactive gene or inactive DNA is preserved in the preservative. It is preserved. Now this is the vaccine. With the preservative, DNA is stored. Now once the DNA of the gene, inactive gene is stored and this DNA is injected into the child's body. Not the virus, here the whole virus, inactive virus, but instead of taking inactive viruses, we are taking inactive gene or inactive DNA fragment that is preserved and injected. Now this, once it is entering inside, it cannot produce a disease. Okay, it cannot produce disease. But the child's body is producing antibody for this. Antibody. So the antibody is ready. Assume that one powerful virus is entering inside. Polio virus is entering. This antibody is going to the virus and inactivating the gene present in the virus. That's all. Poison will not be produced. Even if the virus is alive, the poison will not be produced. Now this type of preserving the DNA of the harmful gene is called, what is that? Present generation vaccine. Next generation vaccine, we can say. That is latest one. We need not think about the whole virus and all. Only one particular gene's DNA we have to preserve. Now that is the third generation vaccine, we can say, or modern vaccine. So these are all the various types of techniques what we are doing. So this is attenuated. That means what? The microorganism is inactivated or the DNA of the microorganism is inactivated. It is called attenuated. Now the DNA vaccine. Now even the whole thing is including everything about the recombinant vaccine. Okay. So that's we have completed about the application of biotechnology in the field of medicine. Okay. The first technology in the first field is over.